Welcome everyone to part 3 of 4 in our Legionella Identification and Prevention webinar series. Today we'll be covering sample collection basics, and my name is Ronnie. I'm the Quality Assurance Manager for ED Lab. Presenting the information for today's webinar with me is Dr. Sahai, the Laboratory Director of ED Lab. Just go ahead and say hello, Dr. Sahai.
Hello, good afternoon, and welcome to all of the participants, and Ronnie, please continue, please.
Right, thank you. So what we're going to cover today, we'll quickly go over a recap of part 1 and part 2. As I said, this is part 3. We also have that information available. If you haven't been a part of this series so far, we can always provide that information for you. We will also be going over the environmental Legionella sampling strategies, process of sampling, and sample transportation. We also have the PowerPoint slides attached to this webinar, so you should be able to download them, but if you don't get a chance to, we can also send you an email for it. Just a quick recap of part 1 and part 2. We went over the 5 W's of Legionella, such as who, what, when, where, and why. The ecology and pathology of Legionella, laboratory techniques, management, regulations, standards, and guidelines, preventative and corrective actions. That was all part of part 1. And then for part 2, we went over some of the core elements of the ASHRAE 188 standard. Here we have the link for the part 1 slides, and like I said, we do have part 2 slides available if anybody needs them. All right, so now we'll go ahead and begin part 3 of our webinar. Dr. Sahai, can you explain what environmental sampling or what environmental samples and sampling are?
Yeah, sure. That's the first thing one should know, because there is definitely a difference between the testing a specimen and sample collected from the clinical specimen versus the environmental specimen. So first thing first, the simple definition of the environmental sample, as you can see on your screen, is that material or information. Again, I emphasize material and or information collected from an environment source to study their characteristic to describe a situation can be termed as environmental sample. What it means that you can collect, as I said, any material or information from air, water, surface. These are the environmental matrix, technically we call it, and we need to study that in order to describe a situation that might demand after analyzing those samples. So those are environmental sample. The process of environmental sample collection is called as environmental sampling. So why these two terms is very important here, because today we will be mostly emphasizing the various technique and procedure for collecting the sample for testing Legionella. So commonly from environment we collect water and air to test the Legionella amplification or Legionella concentration, if any, in that particular matrix. And that's why it was important, I believe, to understand that what is the environmental sample and the environmental sampling procedure, because that it definitely is going to be a little different than collecting the clinical specimen for testing the Legionella. That's why I thought it's very important to understand that.
Yeah, and I think that was a good clarification. Because they are very different in terms of that. So I guess today, in terms of sampling strategies, we're going to go over the motivation, proactive and reactive motivations as to why you would sample for Legionella. We're going to go over the Modus Operandi, the record keeping for those samples that you collect, and once we have those samples, how you ship and handle them.
That's really important, Ronnie, that what motivated to take this important task, like why you are checking or why you are testing or why you want to gather the information on Legionella. What has the situation prompted you to do that?
I think it's a lot of times people are kind of scared when they find out that people have Legionella, or it's typically after the fact that they know it. Are there any other situations or things like that for why you should be testing or sampling?
Sure, that's a great question, and that's why you can see right now on this slide that we have two different options or two different scenarios what motivate people to do it. The first one is proactive and the second one is reactive. Let me first give you some information on the proactive means. The proactive means that like you are pre-conscious. You don't want to run into a problem. So being a good practice, you want to know that the water system around your building is having any Legionella or not. So in other words, you want to know that there is a presence of Legionella around you or there isn't. The second very good idea to make a plan as a part of water management program. If you have a building water management program, you routinely or periodically also keep monitoring and include that as a part of WMP in order to ascertain that whether you have any Legionella or not. And then obviously if you have that information that you are in a better situation, better understanding, and a situation to respond much quicker, faster, and effective should any need arise, meaning that if any kind of like Legionella problem or people start complaining around that building. That's the proactive measure. But what happened when you run into the problem, you miss that first part cannot be retrieved. So you are going in a reactive mode. What it means that you are going to respond. If you have customer or you have a resident or you have a occupant complaining of some Legionella initiated situation become pretty grim. So you start like exploring whether there is really a Legionella or not. That helps. Then responding to the cluster or outbreak, I said you like maybe a localized cluster or maybe the entire neighborhood has having the outbreak. In that situation, we have to work in a reactive mode to get the information as quickly as possible so that the investigator or evaluators are going to determine the source and cause, like where this source, meaning Legionella, is coming and whether that causative outbreak or cluster is associated with or not. So that is very important and valuable information. And obviously, if you have a Legionella, then you should have a Legionella management program under your WMP. WMP is going to be used in this presentation throughout. That stands for Water Management Program, and that is a better responding in case of a reactive mode. That the two motivation I can see people take this important task.
Okay, thank you for that, Dr. Sahai. And with that, we're going to go ahead and present our first question. See if people have been paying attention. Question is, what is the most common type of Legionella sample collected from an environmental source? Options are water, air, both A and B, or none of the above. Go ahead and give everyone about another 5 seconds. And we'll go ahead and close this one. Go ahead and show everybody where we're at. Looks like 73% of you selected the correct answer, which was both A and B. Now we will continue with the presentation. So now we're going to go over the modus operandi. What are some sampling essentials, Dr. Sahai?
Well, that's why this is the core of today's presentation, that we are going to make our viewers aware that it is very important to collect the sample in a way it should be, because the theory is still valid, garbage in, garbage out. So we must give you information in order to collect the sample in a good way so that a valuable information can be obtained from that sample. And especially, we are talking about the microbiology, so we are in hunt of something living, and we don't want to mess with that because we cannot control life, right? So the first thing first, the sample container, which should be used for collecting Legionella specimen, especially from water, must be sterile. When I say sterile, meaning that there should not be any other microbial growth or any other cross growth or contamination, or the bottle or the container which is used for collecting Legionella should be clean as a whole.
Okay. Can we use a swab for sample collecting?
Sure. As the Legionella sample can be collected from the water, surface, or sometime bulk sample also. So of course, swab sample can be used for collecting especially the surface sample. And for those who are not aware that what the surface sample would be in this case, I can give you some example later during this presentation. But one thing I want to emphasize here that the swab must be of polypropylene material. Cotton swab should be avoided because chances of bacterial amplification with the cotton swab and cross contamination chances and other other microbiological issues can be prudent in the cotton sample. So it is always better to use polypropylene sample. And I can also tell you during the presentation that when you have a recent episode of treatment, if you have a known Legionella problem or routinely if you have treated, then we also need some stabilizer in order to get some information. So yes, swab sample can be used for collecting Legionella.
Okay. What are some other things that some people can use for collecting the samples?
Well, the other information which should be very valuable in the Legionella sampling is the amplification of other microbial entities in that particular water system. For an example, if you have a greater count of heterotrophic bacteria in the water sample, chances are getting bacteria Legionella is great in otherwise situation. So one can also collect the sample to do some heterotrophic bacteria count. The other thing come in my head that if you have a protozoa such as amoeba and stuff like that, then that also a supporting factor for the Legionella growth. So to measure these things, it is very important that we should also collect the pH of that water as well as temperature, because these two parameter is going to be a supportive and helpful to ascertain that the other organisms or other situation is not prevailing which support the other microbial growth, because this microbial growth is going to be a great feeder to the Legionella. So you can see that we can use various tool. In this particular slide, you can see that there is a thermometer, pH meter combined. And that's a very handy. It comes very handy, and you can easily obtain the water and measure the temperature and the pH from that water.
Okay. What are a few more things that people can use in the field for sampling?
Sure. You should be well equipped, because the very first thing is that you should be very clearly demarking or identifying your sample. So we must have a legal custody or the different hand where sample pass through from collecting to the testing in the laboratory. Very first thing I can see the importance is something called a chain of custody. It is nothing but it is a document which reveals the chronology of the sample, what, when, where, whom has collected the sample. That kind of information is given there. So you should have those kind of paper handy while you are collecting the sample, A, for tracking the sample, B, to help laboratory to identify or delineate one sample versus other in case of multiple sample collection in one episode. The second thing, which is sort of chain of custody, but it is called as data sheet, or one can use chain of custody as a data sheet for recording the pH and temperature and any other situation or data which deemed necessary for that particular sample. So that is very important and valuable document. Of course, you should have a pen handy, because you don't go on site and you start looking for the pen, because then you will miss the information. So a pen should be handy, and that helps to record the data on the chain of custody or on the data sheet. You should have some office stationery with you, because you never know what kind of situation you will run into. Maybe you have to have multiple chain of custody or multiple data sheet, so you can put together those things. So you require some office stationery such as scissors, staplers, etc.
Okay, and with that, we're going to go ahead on to our second question. Give everyone about 25 seconds to go ahead and answer it. Question saying, what type of swab is good for Legionella sampling from an environmental source? The options are cotton, polypropylene, both A and B, or none of the above. Everyone about another 10 seconds or so. Okay, I'm going to go ahead and close the question. Share the results. And we got 62% of you selecting the right answer. That answer was polypropylene. You want to make sure that that's the kind of swab that you're going to use when collecting a swab sample for Legionella. Now we'll go ahead and continue with the webinar.
Ronnie, as I was describing you, that there is some office stationery you should be definitely carrying with you. But nevertheless, many of the sample collectors forget that they are going to generate some waste. So make sure that you also carry a biohazard waste bag. It should be properly marked because you never know the sample which you are collecting is having some Legionella into it or other concerned pathogenic species or other situation like a biohazard kind of situation. So it is always a good idea to carry some of those red bag. As you can see, I put this is like a little small bag having a biohazard sign so that you can properly dispose those bags as per the local, state or federal regulation. Especially when you are collecting multiple samples in one episode, then you should have something to contain all the collected sample. And the good idea is that if you are collecting a large volume, then you should be carrying a container such as cooler, and especially on wheel, so that you don't have to lift, because this sometimes get very heavy if you are collecting more than 100 or more samples. So it becomes pretty heavy. So I definitely encourage the sampler. They should be carrying cooler or other good container that fit well with the collected samples, so that it can sip and prevent the odds during the sipping.
All right. Should you wear anything specific when sampling?
That's a very great question, Ronnie. As I said earlier, that you never know. These are environmental sample, and many times people are doing in a proactive manner or in reactive manner. So sometimes you have no clue that the sample you are collecting is having Legionella or other pathogenic bacteria into it or not. So not to scare people, but to just a good practices, it is always encouraged to have PPE that stand for, again I report, that stand for personal protective equipment that actually save the collector especially to get any sort of exposure if the organism is in that particular sample which one want to collect. And this is very simple things such as gloves, face respirator. One thing I want to mention that the face respirator must be N95 filtration or better.
Okay. Is there anything else you can do to make sure you're prepared for sample collecting?
Yes, definitely. Because without making a strategy, the sample collection is not going to run smooth, because you will be running a lot of hiccups. So what you need to do in order to avoid those hiccups, if it is possible, then try to procure the map or other graphics and identify the sampling site as well as source, whether you are going to collect specimen from the cooling tower, you are going to collect the specimen from faucet, you are going to collect the specimen from the second floor of the building or first floor of the building or the ground floor, that sort of thing. And then the type of sample, whether you are collecting the water sample, you are collecting the swab sample, meaning the surface sample, or you are just collecting bulk samples such as the soil from a potted plant, etc. And then obviously, if it is possible, then determine the volume beforehand in order to get your logistic correct, so that you should be carrying appropriate sample containers, swab if needed, or any other essential that required for that particular situation in order to collect the sample.
All right, thank you, Dr. Sahai. What is the best way of water sampling from a potable water source?
Well, this is a great question, and this is perhaps most commonly collected sample for Legionella testing. Typically, it is said that you should be collecting 1 liter of water using aseptic technique, and it typically collected from the point of use, such as faucet. As you can see in this slide, all you have to do start running the faucet, put the container, which is a sterile container you are carrying with you, and collect 1 liter of water. If you have a recent episode of treatment or you have recently treated your water, do not, again I underscore, do not forget to put the stabilizer, which is basically the sodium thiosulfate, and their strength is 0.1 N, and it can be requested from the laboratory, or it can be found in the Legionella sampling kit. So once you collect that, please tight the container and make sure that it is not leaking, the caps are properly screwed, and it is just ready to sip. Afterward, you uniquely identify, and you do this practice with, by the way, any kind of sample you are collecting, and then fill out a proper chain of custody in order to record the information so that the laboratory exactly identify, once they give you the result, that from where the sample was coming from or when the sample was submitted for that particular site.
What about non-potable sources such as a cooling tower?
This is really other common type of sample people collect, and that is typically collected from the cooling tower, as you can see here. Again, you should be getting a sterile container, collect 1 liter of water if you can, but a smaller volume up to 250 ml can also be used. Again, use the aseptic technique and do the same practice, tighten the container, prevent the leak and stuff, and identify the sample very uniquely, and fill the chain of custody before you put this in the sampling sipping container.
Okay, and now we're going to go ahead and take a little break and do our third question of the presentation. Which of the following is a PPE? The options are gloves, face respirator, both A and B, or none of the above. Give everyone about 20 seconds. We're going to go ahead and close the poll. And looks like people are finally paying attention. We got 100% of people answering, selecting the right answer of both A and B, gloves and a face respirator are considered PPE. Now we'll go back to the presentation again. Dr. Sahai, are there any other sources you could go over when it comes to water sample collecting?
Exactly. The Legionella sample is not just meant for the water system run in the building. Rather, it can be also collected from in and around the building. Many times, big buildings have fountains and a spa, etc. The pond around them. So, if it is a pond which is decorative, for making the value real estate, up and stuff, you should be also concerned if especially when you have a Legionella episode. So the pond water can also be collected. Well, typically, as you can see on this slide, pond, fountain, spa, etc., 100 to 300 ml of water is also adequate. If you have pond, you can draw 1 liter of water, as you can see in this picture. Just make sure that you are not collecting the sediment. You are just drawing the water from the pond. And again, the same procedure, tighten it, make sure it's not leaking, identify sample very uniquely, and fill the chain of custody appropriately before you ship it to the laboratory. The other sample, Ronnie, is a very important one, is the swab sample, which you asked me earlier. Okay. And I promise that the swab sample can be collected from areas such as aerotas, faucets, shower head, etc. many times you might have noticed that, like, there is a white deposition sort of thing under the shower head, etc. That sample makes a good sample for testing the Legionella. So you can swab the entire surface, making vertical and horizontal strokes, and then very carefully transfer the swab in its original container. Typically, these swabs are moist. If not, then use distilled or aseptic water to moisten it before you make it ready for laboratory transportation. Again, make sure it's not leaking, meaning the swab is tight with its container, uniquely identified, and chain of custody should be appropriately filled. So that is a good swab sample. As you can see this picture, that we have noticed that, like, this particular faucet is having some kind of deposition, so a swab sample is being collected as with the described process which I just said a minute ago.
Can you explain how to collect a biofilm sample?
This is very interesting, Ronnie, because many a time the biofilms are made and go unnoticed, especially if you have a faucet and the faucet joints, if you open it, then there is a washer and other platform like in between the water supply and the faucet head. And nobody pay typically a lot of attention during the housekeeping practice. So that, like, slowly, if the water start leaching with lot of, organic material there, then they are going to form a film with lot of microbes into it. And that's why the simple term, I'm not going into technical detail because it itself is a big class. So biofilm is being formed, and obviously this bacteria can also hide there. So it is a very good idea to open those joints and see that if you look any kind of sediment, which is typically a little bit of gooey, and you can't miss, as you can see in this photo, that there is a biofilm deposition around the neck joint between the faucet and the water supply. So you can very carefully collect the sample by wiping in a circular motion doing the vertical and horizontal stroke, and collect the swab sample again to lift some of the biofilm sample. Place in the container or its original container, and make sure that it's not leaking or it's not, like, having any crack and things like that. Identify it uniquely, fill the chain of custody before you send to the laboratory.
Okay. With that, we're going to go ahead and ask our fourth question. Sodium thiosulfate is used in Legionella sample collection as a what? The options are stabilizer, testing reagent, both A and B, or none of the above. Give everyone about 20 seconds. And we'll go ahead and close the poll. Show the results. All right, we got most people paying good attention. We got 93% selecting the right answer. Now we will go ahead and continue with the webinar, which we're almost finished with. Thank everybody for participating with us.
And this is the very important one, Ronnie. However, this is not very commonly or typically done sample because chances are not that great that the Legionella bacteria come in water, air. Mostly it resides in the environmental water, but sometimes air sample is being made, especially when you have a sprinkler and things like that. And we run the air sample by typically doing the bioaerosol technique, which basically utilizes the adsorption sampler. We use a specific media that is being loved by this Legionella bacteria, such as TSA, BCYE, and flow rate of the pump is need to be 23.1 liter, and the sample should be run a little longer because, as I said, the concentration of bacteria in the air is not that great. So again, you collect the sample by using aseptic technique, by bioaerosol technique or method, and fill out the chain of custody before you submit this sample to the laboratory.
Okay, so now we've gone over many different types of sampling. Now we'll go ahead and talk about the sample transportation. What is required when transporting Legionella samples, Dr.
Well, as you can see, I have listed here very specifically that all the sample must be uniquely identified. A proper chain of custody must accomplish the sample. Sample preservation should be done in a right manner, should be avoiding the excess stream of temperature and the the light, etc. And if sample is required, transportation on the odd condition, I encourage people to do the freeze pack rather ice, and it is always a good idea, and it should be overnight or prior to delivery, and the intent should be putting the sample as quickly as possible to the laboratory. As you can see in the photo, that always provide a thermo seal container so that the sample should be avoiding all those extreme. As you can see, there is some sample inside, and the ice pack and chain of custody, etc., minimum need to be transported to the laboratory at the earliest.
Okay, thank you, Dr. Sahai. And we will go ahead and ask our last question of the presentation. What microbiological media is suitable for Legionella air sampling? Our options are BCYE, MEA, both A and B, or none of the above. Give everyone about another 10 seconds to answer. We will go ahead and close the question now. And share those results. And we have 45% of people selecting BCYE, which is the correct answer. Okay. We want to thank everybody for joining us for this presentation. We hope it was useful for you. And if you need to, check out the previous ones, and hopefully we'll see you here for part 4 of our webinar series. Dr. Sahai, do you want to mention the conference you will be presenting at?
Yeah, I will be in a conference, FRACCA conference, that is scheduled for March 24 at 4 p.m. And I will be presenting there on the Legionella, and we will be getting some information out there. And we can also interact, whosoever is interested, including sample collection to sample testing to interpretation and whole 9 yards on this. So title of the presentation is Legionella Identification and Prevention. So it is very in-depth presentation. If you happens to be in that area or you want to join, please join us. This is in Orlando, Kirby Royal Orlando. This is a hotel there. The conference will be hosted. It's again the FRACCA conference. If you need more information, please shoot us an email. Ronnie is going to show you the contact information, and you can procure more information on that.
Yep, we'll go ahead and leave this slide up for a little bit longer just in case anyone needs it. And once again, we do have these slides available for download, and we will go ahead and send out emails for anyone who needs them. Once again, thank you all for participating with us, and we hope to see you for part 4.
And thank you very much again, and hopefully if you have any other question, you can send us. We can interact. We can give you the information, and we can share our experience and expertise. Thank you very much again for joining us today.